TS Srinivasan Oration by Professor Simon Shorvon. Dr. E.S. Krishnamoorthy will deliver the welcome address.
Professor Simon Shorvon, Mr. Venu Srinivasan, Professor Srinivas, distinguished guests who have assembled here, ladies and gentlemen, and a very special mention of our guests from overseas, experts in epilepsy from around the world. I stand here in front of you to represent both the TS Srinivasan Centre and the VHS. The VHS as an organization is a legacy of a very great man, Professor K.S. Sanjeevi, who had three fundamental principles to healthcare: prevention of disease, which he said was always better than cure; the role of the family as the healthcare provider, which even today in India remains extremely important; and the need for healthcare delivery to be evidence-based and to follow some principles, which is very, very important even today. VHS and the center, the TS Srinivasan Centre, we follow the same principles, and we think of clinical care first, then some teaching, and finally some research. Thinking about this particular oration, which is now entering its fourth decade, the 31st oration, I was wondering, how is it important and how is it relevant? Why does it make a difference to society? And the first thing that strikes me is that these are iconic events, an event that is in its fourth decade, which year on year has brought experts to Indian shores, made knowledge at the cutting edge accessible to a very, very wide audience of people, both specialists and lay people. It's inspirational. It's a symbol of knowledge, of virtue, of philanthropy. And in some ways, there were two famous words that Gorbachev brought out: glasnost and perestroika, openness and progress. It's a symbol of both those important parts of society, openness and progress. And in the imperfect world that we live in, when we are preoccupied with 2G, 3G spectrum, it is a symbol that we have good things happening around us that make life worth living. And I think that that is why programs like this are very, very important. And I would take my hat off to the Srinivasan family for not only conceptualizing this event, but keeping it going into its fourth decade. A brief word about today's topic, heredity. Oliver Wendell Holmes said, "Heredity is an omnibus in which our ancestors ride, and every now and then, one of them puts his head out and embarrasses us." And illness, I suppose, is one such embarrassment. You carry the illness within you, and it expresses itself in your family, in your generation, in a particular way. It's also a reminder of the biological links that we have with our past and with our future, links of which the traces that we cannot actually wipe away. And in the Indian tradition, we refer to karma, the karma that we inherit, and the karma that is our legacy to our progeny. And what's very, very interesting is that when you understand the biology and the genetics of happiness, that thing that we all crave, it is today believed that there is a proximal umbrella of genes, people you are close to, people you are closely associated with during your lifetime, and then a distal umbrella. And the distal umbrella is people you are associated with, people you interact with, people you may help, or people who may help you. And what is becoming obvious is that altruism, which is doing good for many people around you, is passed on down generations. So that's what guarantees the happiness of your progeny. So in a sense, heredity is important, and your actions during this lifetime are important because you will be passing on some goodness to your progeny and to the future generations. So I think that there is a biological link, and I'm sure that's not what we're going to hear about today, but it's an interesting concept. Ralph Waldo Emerson said, "A man finds room in a few square inches of his face for the traits of all his ancestors, for the expression of all his history and all his wants." And heredity, therefore, is a very interesting concept because we see how people inherit features and how when people inherit features, they also perhaps inherit disease. And that is something that I'm sure we'll hear more about today. Finally, one more quotation about poetry and heredity. "Teach your children poetry. It opens the mind. It lends grace to wisdom and makes heroic virtues hereditary." So I think there's much to be said about heredity, and I'm very, very pleased to welcome all of you to this very important oration, which is going to address this very complex issue on heredity. Thank you so much.
The president of the function, Mr. Venu Srinivasan, to deliver his address.
Professor Srinivas, Dr. TS Krishnamoorthy, and our visiting orator, Professor Shorvon, and other distinguished doctors from overseas, members of the audience, ladies and gentlemen. It's always a great pleasure to be here in February when we have this oration. And as Mahesh, as Dr. Krishnamurthy is known, said this is the fourth decade of this oration. I must first thank Professor Srinivas for having really thought about it. It was he who put the idea, the germ of the idea in my mind, and who actively engaged in inviting and getting the leading neurologists of the world to come to India at a time when India was really lacking in overseas connections and education, and start this program of the TS Srinivasan Endowment Lecture. The reason he did it, he was a great friend of my father. My father had a great and abiding interest in medicine, sometimes not always to the good because he experimented upon himself, which I think led to some of his neurological problems, and Dr. Srinivas is closely associated with him. But more importantly, my father greatly cared for society and medical care to the people who were deprived of any access to medical care. He was also a great humanitarian, a philanthropist, and a long term had an association with VHS as well. Dr. Murli, who was the predecessor to Dr. Krishnamoorthy, was a great and close friend of my father and was supported by him in his endeavors. The TS Srinivasan Neurosciences movement began with clinical care in a small primary health center. But slowly, with a long-term vision of excellence and bringing futuristic care to the poor people, this association has grown with this lecture series. The orations have brought some of the most eminent neurologists in the world today. And a few years ago, when it was mentioned who were the members of the Society of Neurology in America, almost all of them who were living had been lecturers in the series. And this year, no less, we have Professor Simon Shorvon, one of the world's leading epilepsy experts in the world. The TS Srinivasan Centre at VHS, over the last few years, under the able leadership of Dr. Krishnamoorthy, plays an active role in delivering multidisciplinary clinical care to over 4,000 patients per year who are treated at the Institute of Neurological Sciences. But most importantly, carefully the data is recorded for clinical study, for epidemiological studies, and it has an active group of professionals working in diverse fields of neuroscience, both clinical practice and in research. Today, it is recognized by the University of Madras to conduct teaching programs as a Master of Science in Neurosciences, an interdisciplinary PhD in neuropsychiatry with the Clinical Neuroscience and Disability Department of Tamil Nadu, Dr. MGR Medical University. There's a fellowship in neuropsychiatry recognized by the university, and this will commence this year starting in July. This is a very unique and first of its kind multidisciplinary fellowship that will train qualified neurologists and psychiatrists to interface in this discipline over 1 year. The TS Srinivasan Centre is the first center in India to offer such a formal university accredited training. A host of scientific meetings that bring international expertise to Indian shores take place under the aegis of the center. Each year, during this lecture, we have, as we have today in front of us, a group of eminent doctors from around the world taking part in this process of discussing and bringing knowledge to India. The development of evidence-based health policy in India and globally is one of the aims of the Center of the TS Srinivasan Department of Neurology. The International League Against Epilepsy, the Indian Epilepsy Association, and the Indian Epilepsy Society has resulted in a development of a novel clinical and health policy concept called disabling epilepsy. The National Brain Research Centre for the Development of Research Protocols and Paradigms on the interface between dementia and cerebrovascular diseases are also there. In addition to that, the Alzheimer's and Related Disorders Society of India for the development of the Dementia India report. So actually the work being done under Dr. Krishnamoorthy has many facets to it, and due to his dynamism, I think has brought this institution to the forefront in neurology in India. This year, the center has started VHS CARES. CARES stands for Center for Advanced Rehabilitation Specialties, and it offers multidisciplinary services again with 38 beds dedicated to rehabilitation and care of the elderly and the disabled. The center caters to all forms of people with disability, step-down care for people recovering from acute medical conditions, respite care for people with chronic disabling conditions like dementia, stroke, and spinal injuries, long-term care for the medically disabled elderly. The project is already functioning in the VHS campus for the last 4 months and will be inaugurated later this year. In 2011, the TS Srinivasan Conclave is a global approach to epilepsy and has been discussed by experts representing the major continents for the last 2 days. The conclave will lead to the production of a book, Epilepsy, a Global Approach, edited by Professors E.S. Krishnamoorthy, Simon Shorvon, and Stephen Schachter of Harvard University. As you'd all remember, Professor Schachter was the orator in 2008. The release in October 10 of the first book to emerge from the conclave, Dementia, a Global Approach, edited by Professors Krishnamoorthy, Martin Price, and Jeffrey Cummings. Again, Professor Jeffrey Cummings was an orator in 2003. Well, this book was a result of the discussion and debate among experts on dementia from around the world. Many staff members of the center have received individual recognition in the last year for their efforts. My congratulations are due to every one of them. We thank the team at the Institute of Neurological Sciences, VHS Hospital, the new and dynamic VHS CARES team, the M.A. Chidambaram School and College of Nursing, and the management of VHS for all the support that the TS Srinivasan Centre and its projects receive. I would now like to introduce our speaker today, Professor Simon Shorvon. He was born in London in a medical family. Both his parents were psychiatrists. He was educated in Cambridge and went on to acquire his MBE, CHB, and MA in Fine Arts there. Following his MRCP, he began his neurological training and soon became interested in epilepsy. He became a senior lecturer and consultant in neurology at the world-renowned Institute of Neurology and National Hospital for Neurology and Neurosurgery, Queen Square, London. He is currently the Professor of Neurology and Clinical Subdean in that institution. He has also served during his long and illustrious career as Medical Director of the world-famous National Society for Epilepsy in the UK. He is currently the editor of Epilepsy, the most important scientific journal in epilepsy, and has been in that position from 2006. Professor Shorvon has received a number of awards for his contribution to epilepsy. Important among these are the European Lifetime Achievement Award in 2008 and the Lennox Award in 2010. He has, to his credit, over 500 scientific articles and book chapters and has written or edited over 15 important books. His contributions to epidemiology, genetics, and emergencies in epilepsy are particularly well known. He is considered an authority in all these areas internationally, and he is married to Lynn, a practicing doctor, and they have a young son as well. Unfortunately, I think we have missed your wife this time. And so with these words, I'd like to introduce Professor Shorvon and thank him on behalf of myself and my family for being here with us today. I'd now like to present the medal and the
Well, ladies and gentlemen, Mr. Srinivasan and Professor Srinivas, it's really an enormous honor for me to be invited to give the Srinivasan Memorial Lecture. I'm told by Krish that one of the lecturers actually collapsed on stage, and so I'm going to do my very best to keep upright. And I hope I'm going to cover this difficult topic, the question of the heredity of epilepsy. And I hope I'm going to speak for about an hour, and I hope I don't bore you too much with this topic. I never met Mr. Srinivasan, but I have grown to know about him from the many dedications and articles which I have read. And I'd like to begin, therefore, first by paying tribute to Mr. TS Srinivasan and also to his extended Srinivasan family. TS Srinivasan had a career of brilliance and innovation. And as Dr. Krishnamoorthy told me, his achievements were also the embodiment of compassion. His was a life devoted to advancement and improvement in industry and also in public service, and not least to neuroscience. I learned a great deal about the generosity of Mr. Srinivasan and his family in the field of neurology, and their support of a number of ventures and developments at the VHS in Chennai. I've, over the past 40 years, visited India on quite a few occasions, and in the last 15 years have been a sort of regular visitor to VHS as a guest of Professor Srinivasan and also his son. And it's extraordinarily gratifying to see the flourishing state of neurological care at the hospital and the clinical work matching, as I think it now does, the best in the world. I was also very impressed on this current visit to see the newly opened Centre for Advanced Rehabilitation Specialties, which was sponsored by TVS Motors, and which is an important step, I believe. I feel humbled by the qualities and example of Mr. TS Srinivasan and his family, but we should celebrate the achievements the family have brought to neurology through their consistent munificence and support. In this lecture, I thought I would attempt these tasks. In the first part of my talk, I'm going to give an historical overview of the major theories of heredity, starting in the year 1860, which was when modern scientific thought in neurology really began, and over the next 100 years. And in the second part of my talk, talk about the last 50 years, which were what one might call the molecular era, in which there have been phenomenal advances in the science of heredity. In the third part of my talk, I will talk about the current state, both the successes and the failures, and briefly in the last part talk about what might be future directions and the ways in which epilepsy genetic studies may develop in the future. And although in general this is going to be a celebration of the extraordinary advances in genetics, I will emphasize that many of these concepts derive from social fashion and social theory as much as from science. And furthermore, some of the theories proved erroneous or are erroneous, and in their turn had major social implications. And I'm going to emphasize both the good and the bad aspects of these social implications. The basic message is science really is never neutral, least of all genetic science. And I hope you will see that having a historical view is important, because there are lessons from history that we should not forget. Now, this is a potentially very large topic, and furthermore, in parts, it's quite technical. And what I'm going to do is try and take as broad a brush approach as possible, but it's not going to be comprehensive, it's not going to be sufficiently explained. And for these inevitable failings, I wish to apologize in advance. So let's start first with the historical overview. Now, for over 2,000 years, ever since the writing of Hippocrates, people have realized that epilepsy was an inherited disease. But it was really only around 1860 that the modern scientific method began to be used and the modern discussion of this topic was initiated. And I think it's important to remember that this was an era when there was no investigatory facilities at all. There were no scans, there was no EEG, and doctors only had the clinical history and the clinical material on which to make a diagnosis. It was indeed a time when even neurology had only just begun to exist as a specialty, and when there was no classification of neurological disease whatsoever. There was also at this time no idea how heredity worked. Although it was recognized that somehow epilepsy was inherited, how this happened was completely unknown. Darwin's Origin of the Species had been published only the year before, and Mendel, who devised his laws, devised these 10 years later. The word genetic and gene were not conceived. And yet, despite this, there were some very interesting concepts of heredity in neurological disease formed at these times, which still have resonance today. Now, the contemporary picture in relation to epilepsy can be gained from 3 books published in 1860, 1861, and 1888. And in these books, there are 4 concepts which I'd like to expand upon. The first was that the universal belief that epilepsy was always due to an interaction between inheritance, inherited factors, and other causes, outside factors, sometimes things like brain tumors or strokes, but sometimes environmental factors. But these factors always interacted with inherited factors. And they divided these causes into 2, what they called the predisposing causes, which was inheritance, and the exciting causes, which were these other factors. And Sievking, who always wrote in a very flowery and imaginative way, thought of it like gunpowder and a match. The gunpowder was the inheritance. The drier the gunpowder, the more likely it was to explode. And the smaller and the drier it was, the smaller was needed to be the exciting factor. And this idea that there is a mixture of factors is still very important today, and we'll discuss that a bit later. Russell Reynolds considered hereditary to be the major influence in 30% of his cases, and Gowers, William Gowers, of course, who was the renowned English neurologist, thought hereditary was the overwhelming cause in over 60% of his cases. The greatest neurologist of the time, of course, was John Hewlings Jackson. And he had some very interesting ideas on causation. He accepted that there were predisposing and exciting causes. And he also accepted the predisposing cause was inherited. And his great contribution was to recognize that the inherited mechanism was hyperexcitability of the cortex. This was an amazing insight, really, at a time when there was no chemistry and there was no physiology. And he realized that the hyperexcitability was, in fact, in his view, inherited. He interestingly had no interest in other causes and thought the cause of epilepsy was the mechanism of epilepsy. And that's a very important concept, which again, I think only recently people are again focusing upon. He thought that all seizures, whatever their cause, had this final common pathway, which was overexcitability of nervous tissue, in his view, due to deranged nutrition. But this overexcitability was the cause, whatever was the more downstream influence, causes and influence. The third concept, which was very important in those days, was the concept of the neurological trait. Now, this is an interesting idea which had developed in the mid-century sometime, but by 1880, in the time that Gowers wrote, it was universally accepted. It's hardly discussed now until in recent years, until very recently, when there seem to be modern genetic concepts reinventing this idea. According to this theory, individuals didn't inherit epilepsy alone. They inherited a range of neurological conditions, of which epilepsy was only one. This inherited propensity was known as the neurological trait, and it contained various conditions, including some psychiatric disorders, sometimes other neurological disorders, some mental retardation, and also, interestingly, some personality disorders. And as Gowers wrote, it is well known that the neuropathic tendency does not always manifest itself in the same form. the chief morbid states beside epilepsy itself, by which the same tendency is manifest, is insanity, and to a much smaller degree, other chronic forms of disease of the brain and of the spinal cord. And intemperance, too, is probably due, in many cases, to the same disposition. That's a thought, that's an idea we'll come back to later. Now, the fourth concept I want to mention, which was of great importance, and which proved to be completely erroneous, and that is the theory of degeneration. I'm going to mention it now because of the historical consequences which it had and the lessons from which we might learn from these events. Now, the theory of degeneration was originated outside medicine, more in many other types of social discourse, not least in population science. And according to the theory, there was an inherited propensity in some individuals for progressive degeneration, which became more severe as generations moved with each generation. So, for instance, and epilepsy was thought by many to be at the core of this tendency. So, for instance, one might have migraine in one generation, then in the next generation you might get epilepsy, in the next generation you may get dementia, and so on, and eventually the line would become extinct. According to these theories, degeneration brought out what were called atavistic traits, which were primitive traits which had disappeared during evolution, and which didn't manifest except in these unfortunate individuals exhibiting these traits. They didn't manifest themselves, but they were present in the germline, although not expressed in most individuals. This theory was a social theory too, as I will explain later. Now, this theory of inherited degeneration reached its peak at about the end of the 19th century with the work of Cesare Lombroso, who was not a physician but a criminologist. And his experimental method was to measure, to look at inherited characteristics, facial characteristics, bodily characteristics, and also psychological characteristics. And he made the extraordinary step in considering that criminality in 2/3 of patients was an inherited trait, and that criminals weren't made, but people didn't become criminals, but they possessed a criminal trait, and they were what he called born criminals, and that criminality was one of these atavistic throwbacks to previous stages in evolution. Now, this had come at a time when people were looking at populations too, and there was great anxiety in the European populations that with the move of the classes from the rural areas into towns, with people, the lower classes, breeding faster than the upper classes, that the population was beginning to be weakened. And in Britain, when the Boer War came and the British Army performed very badly, there was a lot of discussion that this was because of the general weakening of the population. And so Lombroso's ideas came on fertile ground. And I mention these because these are examples of these medical ideas getting into social fashion, and social fashion influencing medicine. And then in the 4th edition of his book, Lombroso made the really disastrous link between epilepsy and criminality. And he proposed that epilepsy itself was an atavistic characteristic and was a fundamental component of the criminal type. And he supported this by showing pictures of patients, people with epilepsy, and pictures of people with criminal pictures, people from prisons, and making various measurements and so on, he found that they shared similar characteristics. And indeed, he wrote that 27% of all epileptics, men, and 25% of all epileptic women had the full criminal type. Now, this is obviously completely wrong, and the theory has no basis in scientific fact. But it does, in my mind, illustrate 3 points which are interesting and valuable today. The first is that medical theories often emerge from the wider social context. As I've said, the theories of degeneration arose because of ideas of degeneration in population, and also the arts in those days were full of pictures of arts and literature, full of pictures of people with degenerative tendencies. The point was to emphasize that the scientific theories mirrored contemporary social theory, and that they were not really objective. And we like to think nowadays of science being objective, but one should recognize that that's not always the case. The second is that although they're clearly wrong and very biased, these theories were universally accepted by doctors and the profession, and indeed most scientists. And I think it demonstrates again how skeptical we should be of all scientific theory. And the final point is that the association of epilepsy with this whole business of degeneration became immensely stigmatizing. And this opened the gate for the next scientific development, which was to prove truly disastrous in relation to epilepsy. And this, of course, was the science of eugenics. Now, eugenics became the predominant hereditarian theory in the first 3 decades of the last century. Eugenics was the science of improving genetic qualities in a population by selective breeding. It developed from Darwinian theories of natural selection and survival. It was a term coined by Galton, who advocated positive eugenic solutions. In other words, not negative solutions, which I'll come on to, but positive solutions. And he wasn't interested in medicine. He was much more interested in populations. Now, medical diseases became the subject of eugenic study in about 1900. And epilepsy, because of its link to degeneration and criminality, was soon the focus and the central feature of many of these studies. And a research office was set up in Woods Hole in the United States with Charles Davenport as director. And in 1911, Davenport produced a seminal paper in which he purported to show that epilepsy was a recessive disorder inherited along with imbecility and with negative psychiatric features. Now, these are his family trees. This was his paper. There's been a lot of studies since shown that, in fact, he simplified a lot of this data, and in fact may even have fabricated some of his data. And certainly his idea that epilepsy was a recessive disorder is completely wrong. Nevertheless, it had enormous consequences, because soon after this, the idea arose, the eugenic idea arose, that if you somehow stopped people with epilepsy reproducing, then you might cut down the number of cases by selecting out the genetic disorder. Now, of course, the genetics, this is completely wrong from the point of view of modern genetics. That's not how the condition is inherited. But nevertheless, compulsory sterilization started and was soon adopted in 23 states. And I'm interested that Krish quoted Oliver Wendell Holmes, who was the Chief Justice at the time in the Supreme Court in America, because he made this statement in this extraordinary case of Bell versus Buck, where Carrie Buck was a lady with a mental retardation and epilepsy. She was forcibly sterilized. She appealed, and Wendell Holmes said it was better for all the world instead of waiting to execute degenerate offspring for crime or let them starve for their imbecility, society should prevent those who are manifestly unfit from continuing their kind, and he recommended her compulsory sterilization. It turned out in the end that she wasn't even mentally handicapped, and this was a notorious case, but which opened the floodgates for enforced sterilization. Now, of course, this all ended in disaster. With the Nazi periods, eugenics became the justification for the mass murder of the mentally handicapped and the unfit, including those with epilepsy. And it was sanctioned under the guise that this was mercy killing, which would prevent the population from becoming weakened by these people. So what happened was physicians provided the names, and the victims were either gassed or poisoned. And it's been estimated that up to 700,000 hopeless cases who were living in institutions were murdered in this way. Now, when all this came to light after the war, the science of genetics in epilepsy basically became out of bounds. And for the next 20 or 30 years, very little activity occurred. This brings us really to the end of this historical period, which was in 1960, with the famous American neurologist William Lennox, who was interested in heredity, and he made one conspicuous contribution, which was the idea, reinventing really the idea, that genetics interacted with other factors to create epilepsy. And he had this very colorful concept of streams running into a river, the river being the patient with epilepsy, the streams being the various influences which lead to the condition. So some of them were genetic, some were acquired, some were environmental, and some were hereditary. And then the stream runs into a reservoir, and the height of the banks of the reservoir determine whether or not the person has a seizure, and the banks being set largely by genetics, but also by environmental causes. And this was a very colorful way of expressing what was really known about the heredity of epilepsy in about 1960. Now this brings us then to the next phase, which is the era of molecular genetics. There's a picture here of the human chromosomes, 46 of them. In fact, this number was only really fully understood to be the number in 1956. It seems an extraordinarily short time ago. And in 1953, the amazing discoveries of how genetics operated were made by Watson and Crick. They discovered that although it was known that heredity somehow inherited characteristics were somehow passed by the nucleus of a cell and probably by DNA, it wasn't realized how the whole thing was coded. And Watson and Crick devised the model of the double helix, which I guess everybody here has heard about, in which there are chains of nucleotide bases, 4 types of bases, which code for proteins. And these chains, 3 of these bases, form a codon. There are 4 different types of bases. You can have many combinations, 20 or more combinations. They code for an amino acid. The RNA copies from the DNA, which is then transfers the codon into an amino acid. And a protein is built up from a collection of amino acids. So the string of DNA and base codes are translated into strings of amino acids, which form the protein. And variations in the genetic code end up as variations in protein. And these variations sometimes cause disease. And so what you're inheriting here from this code, transferred into the protein, transferred into the body, causes the disease. Now, after this discovery was made, there was really rapid progress. By 1972, the first whole genome, the whole genetic code of an organism was published. And then the invention of DNA sequencing by Sanger was invented. In other words, a method for identifying the code. Then it was realized that there were some genes, also things called regulator genes. And by the 1970s, it was realized that they were getting close to the whole human genome. And in 2000, the first whole genome, the first whole code of a human person was published. We recognized a little bit earlier that the human and the chimpanzee differed by only 1%. And in fact, the human differs from the earthworm, really. They share 75% of genes. And one of the enormous surprises of the Genome Project, really, was how few genes there are. And there are only 20 to 12,000, 25,000 in the human genome. And so the whole genetic basis, with all the variation of human and indeed the animal kingdom, is based on these genes. And very soon we'll have the human genome for $1,000, and all of us can get our genome identified and published. So where does epilepsy fit into all this? Sorry, could you move the slide up? Maybe the... Thank you. Okay. Well, in these 50 years, there have been a number of major advances. And I think in the field of epilepsy, I've listed these here. First, all the Mendelian disorders, the genetic disorders, the single gene disorders, in which epilepsy is part of the phenotype, part of the overall clinical picture, pretty well all of them have now been identified. There are over 200, and they've all been discovered by what are called family linkage studies, where you have a family with lots of members with these conditions, and you compare the genome of the members with the condition with the genome of members without the condition, basically, and you can identify the genetic defect. Now, out of these 200 or so cases, 200 and so conditions, 13 genes were found where the persons only had epilepsy, but they didn't have any other features. And these have been called the pure epilepsies. The first one was discovered in Melbourne in 1994, and since then there have been 13 genes identified, defects of which cause pure epilepsy without all the other features of mental retardation or other conditions. And as I say, all of them have been discovered by family linkage studies. These are the 13 genes. These are the epilepsy conditions which they produce. There are two key points. The first is that these conditions are generally quite rare. And these 13 genes account for a very small number of the overall population of epilepsy. 1 in 200 people in the population have epilepsy. About 1% of those 1 in 200 has defects in one of these genes. And the second interesting feature is the same gene can produce different types of epilepsy syndromes. And that immediately tells you that it's not just a simple transcription. There must be some other complicated feature. The second fact for these are that all these genes turn out to be what are called ion channel genes. They're genes which code for a protein in the membrane of neurons. And this was an enormous shock. And for the first time, it was then postulated that epilepsy might be what's called a channelopathy, a disease of ion channels. In the meantime, enormous progress was made in the molecular basis of the membranes. And indeed, these channels, which are units which are put into the membranes, the long neuronal membranes, and which allow the passage of ions in and out of the cell. And it's the passage of these ions which changes the electrical characteristic of the cell, which allows a nerve discharge to pass along the axon. These have now been identified in 3D by crystallography, and the whole genetic basis of each of these channel genes, the whole genome structure has been identified. And defects are shown in this picture where little defects in various parts of these genes result in a change in the structure of the protein, which results in a deficiency in the passage of the ion, either too much or too little. And it's that changes the electrical characteristic of the cell, and it's the change in electrical characteristic cell which causes hyperexcitability, as Jackson put it, and which makes the cell fire off in an epileptic fashion. So here's an example where the genetics has led to an understanding of the mechanism, which has led to a plausible explanation of why a seizure occurs. And you can look at the electrical patterns of cells with defects in these genes compared with cells which are normal. And you can see their electrical pattern differs, and the way that spontaneous firing occurs in those cells also differs on the basis of these channel genes. So this was an extraordinary development. It showed that epilepsy, there were 13 genes which were found which underlay some forms of epilepsy. These were all channel genes. This affected the electrical nature of the membrane, and that you can see how that electrical disturbance immediately could translate into an epileptic seizure. And so this was a phenomenal development. Having done this, people started looking at the common epilepsies, not the ones running in families where you could do a linkage study, but they just looked at random patients with epilepsy and carried out what are called case control association studies. And this is one of the great disappointments of modern genetics in epilepsy, that these case control association studies have led absolutely nowhere. And this was a study again from Australia showing that 165 have been carried out, $100 billion spent on these, and we have 0 results. I think what it does show is that epilepsy is a complicated affair, and that except in these families, which are very exceptional, in the ordinary person with epilepsy, although there may well be a genetic basis, it's not the result of a single gene mutation. And as Steve Jones, who's a very famous biologist in London, wrote in the British Medical Journal, the mountain has labored and brought forth a mouse. And it's very difficult to disagree with this, which is one of the major disappointments of genetics in recent years. Now, so what about the future? That's pretty well where we are now. And I'm going to end this lecture by considering a couple of examples of the complexity which exists in the genetics of epilepsy, which may help explain the paradox that although everybody agrees epilepsy must have a genetic basis, the genetic form of this basis is largely still completely undiscovered. Now, there's a couple of clues, and this is some interesting recent work which has not looked at defects in individual genes, but has looked at what are called copy number variants, which are the presence in the genetic code of duplications of genes, or sometimes deletions of bits of genes, or even whole genes. And these duplications or deletions, which sometimes occur as the thing is being copied, anybody who's ever done any massive photocopying will know that you occasionally make mistakes. Well, so does the copying of DNA. And sometimes you miss a bit, or sometimes you duplicate a bit. And And people have been looking at the presence of copy number variations in epilepsy, and they've been finding that some patients with epilepsy have these copy number variations. That's discovery number one. But the second most intriguing finding, really, is that it is the same copy number variants that underpin epilepsy as also underpin some of the neuropsychiatric disorders which had been in parallel studied. So, for instance, the first one found was a deletion there, which is also found in patients with schizophrenia. Then a large study was carried out finding deletions and duplications at 5 sites on the genome. And some of these sites are shared with patients who also have autism, different patients who suffer from autism. This really, to me, seems to be a reinvention of the idea that epilepsy is inherited by mechanisms by which other neurological diseases are also inherited, including some of the neuropsychiatric disorders, which is, of course, exactly what Gowers and co. were saying in the middle of the 19th century. Now, we've been for some years looking at what's called comorbidity, in which is the fact that people with epilepsy tend to have more depression and sometimes other problems associated with the epilepsy. And it has been up till now generally assumed that comorbidity was caused by having epilepsy. But now it looks as if it's possible that these associations are not causal at all, but they reflect the fact that both may have the same mechanism. And in fact, people have now been looking at whether comorbidities predate the epilepsy and therefore can't be due to the epilepsy. And in fact, it's been found that the incidence of depression, for instance, and psychosis in patients who subsequently develop epilepsy is a lot higher than it would be in a controlled population also. And so the so-called bidirectionality of comorbidity may be something to do with the shared genetic basis. At least that seems to be a hypothesis which would be well worth following up. Also found was using these studies of copy number variations, people have found that the expression of a gene which is affected by, say, a deletion, the expression of that gene is affected, but also a whole lot of other genes around it also seem to be affected. And there are now beginning to be recognized various ways in which this may occur. And this finding of copy number variation also reveals the potential for what might be called reverse phenotyping, where instead of looking at a person with epilepsy and trying to find the genetic basis, That's obviously a very important question. I think the main impact to date of genetics has been, is this working? Has been in the field, for instance, of cancer. Shall I speak now? Yeah. I think the main implications of genetics to date have been in the field of cancer and, say, immunology. And it does seem to be that in these fields, single gene defects can have a major effect. Epilepsy being a developmental disorder or a degenerative disorder possibly is more complex, and it does seem that in the neurology disorders anyway, it's not only epilepsy, other neurological conditions, the impact has been less. And I think that may reflect the nature of the abnormality, which in cancer may be a single breakpoint in a single gene. That's definitely not the case in neurology, in most, anyway, neurological conditions. And as for the question of personalized medicine, a lot has been said. I remember going to a meeting about 10 years ago in London where the head of Glaxo came along and said, we must all give him all our patients' DNA, and within a few years, there will be personalized treatment of epilepsy. Well, it has simply not happened. And if you ask me, the reason it's not happened is that there are so many factors which influence response to treatment, which have absolutely nothing to do with the genetics of an individual, that it would be, in a way, foolish to expect it to have any difference. So, for instance, in epilepsy, the type of epilepsy, the severity, if you've got a lesion, the position in the brain of the lesion, the size of the lesion, if you've got a drug, the dose of the drug, all of these influence. We all know people have seizures when they have too much alcohol, too little sleep. None of this is genetic. It may have some sort of genetic implication, but it's very unlikely to be a single genetic factor. And because of that, my personal view is that I live to be proved completely wrong, but I suspect that we're never going to identify strong genetic influences which affect response to treatment.
Doctor, may I have a mic? You raised one particular issue which is of concern to society and has some serious consequences of way insurance is given. And your mention of creating disabled minorities. And as you link cancer or some other diseases, maybe cardiac, diabetes, as is known, how does the governments view DNA studies and the way insurance or care is then becomes available to the average citizen?
It's an extremely complicated situation. I suppose a lot comes down to the reason for people looking into this in government or anything else. I mean, if it is a beneficent reason, I mean, if you want to try to identify the cancer genes so that you can develop therapy for that genetic defect, that's obviously fine. If, on the other hand, you want to identify cancer genes so that you exclude these people from the breeding population or something, which is exactly what happened in eugenics, then that is a great mistake. It was a great mistake in eugenics because it was based on completely erroneous ideas on how genetic influences were in fact inherited. They're not inherited simply down a single line. It's much more complicated than that. It's interactions, it's groups of genes, it's genes plus environment. So I think for certain diseases, there may be reasons for governments and authorities to have information, but for other situations, I would suggest that science try to put a ring around the usage of this information.
Yes.
During the course of your speech, you had pointed out that a nervous disorder was due to distorted nutrition. And then you went on to say that distorted nutrition was due to vascular congestions. So it clearly hammers a point that everything is due to heredity factor. The point number two is, then you went on to explain that heredity is one of the cause and the acquired syndromes, that is your work environment, could cause neurological disorders. But then when you say about this entire thing is due to your acquired heredity, so the acquired factors does not find a place at all, because we can definitely come to the conclusion that everything is due to heredity. And before that, a small question. And another point number two is, you said 20,000 to 25,000 genes in human DNA. I would just like to find out from you whether any gene therapy treatment is in the offing for curing epilepsy or other neurological disorders.
Okay. The first point about it wasn't me who suggested, this was Hewlings-Jackson 100 years ago, who said, 150 years ago, who said that there were common mechanisms at the end of causing diseases. It didn't matter what influences were coming in, some were environment, some were genetics, head injury, heat, stress. Always there was still the common mechanism, and that was due to a nutritional defect. Of course, he didn't know any idea of chemistry or anything, but in fact, I don't think people now call it nutritional, but they do call it neurochemical. And if you transplant the word neurochemistry for nutrition in Jackson's work, you could almost be looking at something today. And I think the idea that there are a few common pathways probably is correct. Now, the question of gene therapy, if there is a single gene and it explains the whole disease, and that is the situation, say, in some muscle diseases, one or two rare muscle diseases, then somehow putting back that gene might somehow create the normal protein instead of the abnormal protein, and then the organ, the muscle, might begin to work properly. That's the theory. And indeed, in muscle disease, there have been one or two attempts at this. In the more complex central nervous system diseases, I think we are miles away from any genetic therapy, I must say. Again, I look forward to being proved wrong, but I can't personally see there being the slightest chance of a gene therapy in my lifetime anyway, for any of the brain